So hello everyone. So today we have another edition of the ERGA's Data Analysis Committee seminar series, and we are pleased to welcome three speakers today. Whose work spans from evolutionary genomics, quantitative genomics, and conservation genomics. So starting from the first person who will speak, so Hernán Morales is the leader of the Evolutionary and Conservation Genetics Group at the University of Copenhagen. He has a PhD from the Monash University in Australia, and his research focuses on the application of paleogenomics and genomics, quantitative genomics, and evolutionary modeling methods to explore the mechanics behind the generation and preservation and loss of biodiversity. So our second speaker will be Samuel Speak. He is a postdoctoral research scientist at the Harlem Institute, where he studies genetic traits related to environmental stress. His PhD at the University of East Anglia focused on conservation genomics in endangered zoo house birds, evaluating the genetic load of individuals in captive breeding using ultra-conserved genomic elements. And Xuejing Wang, I hope I'm saying your name right, is a postdoctoral researcher in the Evolutionary and Conservation Genomics group at the University of Copenhagen. Her work focuses on endangered birds species using genomic tools to investigate demographic history, genetic load, and temporal dynamics. She's completed a PhD at the University of Bern, working on genomics and evolution of insular mammals. So thank you all three to us for accepting the invitation to present your work for the ERGA and the BGE communities. We look forward to hearing your presentations, and a reminder for people attending broadcasting this on YouTube, you can post your questions on the YouTube chat, and we will read them for our speakers. So please, Hernán, go ahead.
Yes, thank you, Joao. Can you see my screen?
Yes, you can.
Super.
Yes.
Okay, thank you so much, Joao, for the nice presentation. Also, thank you, Maria Jose Ruiz, for the invitation, and Luisa Marins for coordinating this seminar. We're very excited to be here. Let me just minimize this. Good. So yeah, as Joao mentioned, we are gonna have three talks discussing our work on the evolutionary dynamics of genomic erosion. I'm first trying to offer a little bit of an overview of our work and a little bit of background information. And I will very much focus on how we use temporal genomics to study genetic diversity loss over time. So Joao already did a great job at introducing our group. We're a recent formed group. We're about 2 years old. We try to interact with a lot of conservation stakeholders like the Mauritian Wildlife Foundation, the International Crane Foundation, the Difficult Bird Research Group in Australia, ESAS, and also the UCN, to try to make our work a little bit more applicable. But we also collaborate a lot with the Earth BioGenome project, through the Vertebrate Genome project, also do the Darwin Tree of Life, and hopefully as we work with more European species, also with ERGA. So the motivation of our work is that, as we all know, biodiversity is in fast decline around the world. The Living Planet report says track about, I think, 5,000 populations of animals, and they report about an average of 73% reduction in abundance for these populations. Also, there's been a very substantial and really nice recent meta-analysis published in Nature this year, showing that within population genetic diversity is already impacted worldwide in a timescale that is consistent with human activities. On the other hand, we are very happy to see that conservation truly works. There is several reports, but this is a very nice one that showing that conservation work has reduced extinction rates. So essentially, conservation saves species from extinction. So we're wondering what are the long-term consequences of this genetic diversity loss and the interaction with conservation actions. One of our species shows this legacy of population decline very clearly. The pink pigeon in Mauritius declined to horribly low numbers, about 12 to 20 individuals in the late 80s, and it was very successfully recovered demographically from this near extinction event by very intensive conservation action. It was so successful, the conservation action, that it was actually downlisted in the red list twice, from critically endangered to endangered and then done vulnerable. So it's considered to be less threatened than before because of the demographic recovery. However, we also track genetic diversity through time. And at the same time that they were having this fast demographic recovery, they are having this sustained genetic diversity loss because they are suffering from this genetic drift that imposed by long-term consequences of the bottleneck. So we're essentially interested in unraveling the consequences of this genetic diversity loss. And this can be captured as this effect that is the genomic erosion, that is the effect that genetic pools suffer through the genetic diversity loss. It can be expressed in different ways. One is the neutral, genome-wide neutral diversity loss that reduces the adaptive potential of a species to future environmental change, but they can also lose diversity that is useful now. Imagine beneficial variation that is compromised, like loss of immunogenetic diversity. And they also start expressing more the effects, the damaging effects of deleterious variation. And this can cause inbreeding depression and eventually reduce the viability of populations. So we're interested in unraveling these dynamics of genomic erosion. And for that, we use whole genome sequencing. And we try to use different metrics that approximate these different slices of genomic erosion in different ways. For example, genome-wide genetic diversity, immunogenetic diversity, inbreeding through runs of homozygosity, and genetic load. I'm gonna quickly explain a couple of these methods that we use. A few years ago, we put out this review, explain our perspective of how genomics can be used to approximate the genetic load in individuals, essentially how to score deleterious mutations. Our aim here was a little bit as well to unify different definitions of load, because as you can see in the left box, there is many definitions of load and people use them differently. And we try to unify these different definitions in two components of the genetic load. So the genetic load is all the additive effect of this deleterious variation, and it can be divided into the realized load and the mask load. And essentially, this also has an important component for how genetic load operates through time, because realized load is the deleterious variation that is expressed in the present as negative fitness effects. This would be homozygous variation. There's no way to hide, so it's going to fully express, but also partially recessive mutations that express some of the deleterious effects. And then you have the mask load, for example, recessive heterozygous deleterious variation that doesn't express the deleterious effects now in the present, but it can express potential negative effects in the future. For example, if populations go through bottlenecks. We also use these runs of homozygosity that are very useful to track inbreeding through time, but they are also related to this genetic load and expression of the deleterious variation. Here you can see on the left how when you have breeding between two closely related individuals, they will inherit the same genetic tracts, which will generate runs of homozygosity, and then it's likely that the deleterious variation will express in these runs of homozygosity that are deleterious mutations turned into homozygous state. The other useful thing of runs of homozygosity is that they are sort of a record of demographic events, because once formed, recombination will break them down and they will become progressively smaller over time. So we can track a little bit the history of inbreeding, if you want, or the coalescent events created by drift and inbreeding. The other tool that we use is museomics, or the extraction of genomic data from natural history collections. I will not get too deep into this, but it has some particular methods that you need to account for when dealing with this type of data. I'm just gonna do a small plug of a online course that we have coming up later this year as part of the Fisale courses that we're gonna be running at the end of this year. So if you're interested in how to analyze genomic data from natural history collections, you can check it out. So now a little bit the project that we've been running for a number of years now. We're interested in these signatures of temporal genomic erosion for heavily bottleneck species. I'm gonna really quickly mention a few examples of 2 or 3 species. I'm just keeping track of time. And we want to eventually do a comparative genomic approach of different types of bottlenecks, different speeds of bottlenecks, and species that recover and species that do not recover. So I'm gonna walk you through 3 different examples very quickly just to show the type of signatures that we get. The Seychelles paradise flycatcher went down to 28 individuals in the 60s and recovered very successfully demographically. It was downlisted. But in the background of this recovery, it lost about 90% of their historical genetic diversity. And they have these relatively small rows of homozygosity, which are evidence of this historical inbreeding that they suffered in the 60s. But since then, they have broken down these runs of homozygosity. They don't have really, really long runs of homozygosity. That means that inbreeding doesn't operate in very recent times. And this is probably because despite being socially monogamous, they have 71% extra paternity. So there is a lot of like crossbreeding between pairs. But they show accumulation of mildly deleterious variation. So this means that the modern population is less fit than the historical population. So this is something that we can extract with these temporal genomes. Another neat example that was published earlier this year, Luke Silver from Sydney University analyzed the orange-bellied parrot, this heavily endangered parrot in Australia that went down to 13 individuals in 2016. And since the bottleneck is being become susceptible to a viral disease that affects parrots, we recorded 62% of loss of genome-wide diversity. And importantly, we also recorded a lot of loss of alleles in toll-like receptors that are part of the immune response that parrots can mount for this disease. So there is clear link here of loss of immunogenetic diversity and susceptibility to diseases after bottlenecks that we can study with these temporal genomic approaches. And finally, another thing that I want to highlight is how genetic diversity, at least in the way of measure as genome-wide diversity, is slow to respond to population bottlenecks. So we have this regent honeyeater in Australia again, very, very large ancestral population, probably hundreds of thousands of individuals, that went down to 250 individuals in the last few decades. So this is a 99% demographic loss in the species. But we only see about 9% of genome-wide diversity loss. This is because alleles don't fully coalesce yet. We cannot track this with genome-wide metrics. But despite this, we did simulations, and we know that there is genomic erosion already ongoing in this system. So with this little genetic diversity loss doesn't mean that they are not in genetic risk of genomic erosion. We also did some species distribution modeling, and the future doesn't look promising for this species. So this is going to compound the effects of genomic erosion in the near future, and this species is really, really in trouble. So lastly, just to show that we do a lot of simulations of this genomic erosion through time. We use forward in time individual-based simulators for this. And we can vary things like the ancestral population size, and we can vary things like environmental optimums. We can simulate deleterious variation, and we can simulate polygenic traits adapted to these environmental optimums. I'm kind of running out of time, so I will leave the references up there for you to see. But we have seen that the ancestral population size has a strong effect on extinction risk of species through bottlenecks. But depending if we are talking about deleterious variation or polygenic variation adapted to environmental traits. So to summarize, genomic erosion has a time lag, and it's also sustained after population recovery. So this highlights the importance of monitoring genetic diversity through time and not only going by demographic parameters when doing conservation assessments. But also, there is several blind spots that remain in our understanding genomic erosion that I will be very excited to discuss here if anyone is working in these aspects like structural variation or plasticity or compensatory mutations or epistasis. Lots and lots of people to thank, especially my team here in Copenhagen and Sam and Xuejing especially that are following this talk. So in the interest of time, I'm going to leave it there and leave the rest for Sam and Xuejing. Thank you.
Okay, Samuel, I think it's your turn. You turn on your mic.
Hi, can everyone hear me and see me?
Yeah, yes, yeah.
My presentation. That's amazing. Hi. Yeah, thank you, Hernán. That was a really great introduction. As everyone was saying, I did my PhD at the University of East Anglia, focusing on genetic load within captive bred populations with cockroach and Hernán. As Hernán summarized there, genetic load is a big problem for a lot of threatened species. One of the major problems we have is how do you quantify what mutations are actually deleterious and what individuals possess these deleterious mutations and how we can then make those comparisons between individuals to see which are the fittest individuals in the population. So a lot of data exists, not for threatened species, but for model species to be able to assess this information. So my work focused on using this information for model species and lifting it over to threatened species. So to give you a quick bit of background, so one of these mutation impact scores is called Combined Annotation Dependent Depletion Scores, or CAD scores. So these are a way to estimate the impact of how deleterious a mutation is going to be in a genome. So this takes in a lot of surrounding sequence context, evolution constraints to produce this single sort of ranked per base CAD score. So these were developed originally in the human reference genome, so taking around 8.6 billion single nucleotide variants and scoring every single one of them differently across the entire human genome. So I said these create a single ranked per base CAD score. So for every single mutation. So this means that a mutation from an A to a T is going to be different from an A to a C or an A to a G. And it matters where in the genome that mutation is going to be. So the 10% most deleterious mutations get a CAD score of 10 or over. 0.1%, the 1% most are 20 and over, and 0.1 is 30 and over. So as I said, these were originally developed in the human genome, but then they were also applied to other model species. And so the mouse, the pig, and the chicken, which is what I used for my work on the pink pigeon and on the whooping crane. So as I've said, we're able to get these CAD scores that can tell you how deleterious a mutation is, but we wanted to make sure we were identifying truly deleterious mutations that are going to be bad, not just in the organism they were designed in, but also within the species of interest that we're looking at, this threatened species. So a way to do that is to assess in what are called ultra-conserved elements, or UCEs. So these are very small sections of the genome that are highly conserved through time, so not just between pigeons or between pigeons to the chicken, but between all birds and then back to reptiles, for example. And as you can see here from this little sequence alignment, where I've got the chicken at the top, the humans and the pig, these three sequences are highly aligned for that UCE region of just 120 base pairs. But as you get up or downstream of that, the alignment gets less good and you get more gaps appearing. So what I did was I extracted the CAD scores for all of these UCE regions for all these three model species. As you can see, running just along this is just one single UCE region and its flanking regions. As you go along the flanking regions, the CAD scores are fairly low, and as they get up to that UCE in the light gray, they increase and they go back down again. And there's a gene in this downstream flanking region, and you can see that splits out. We find the same signature in all three of these model species. So when we looked at this across all of the UCEs that are shared by all these three species, we could see there was high similarity between the CAD scores. There was a strong correlation between a CAD score in one species to a CAD score in another. So although the individual physical score won't be the same between the two species, if you get a high CAD score in one species, it's likely that it will also have a high CAD score in the UC of another species. So this allowed us to see that this was a good proxy for how deleterious a mutation is, and we can use this for threatened species. And this is all available in this preprint here. So this comes to the main part of my PhD, which was published in this load lift pipeline, which is available in this paper that we published earlier last year in Molecular Resources. So this is the load lift pipeline. So this requires you to take the inputs of those UC probes that you want to investigate, the reference genome for your species of interest, and the reference genome for the species that you have, the model species that you're lifting the CAD scores from, as well as the CAD scores themselves and your read data for your individuals. So what you do is you extract those UC regions from your reference genome, and at the same time you align that reference genome to the reference genome for your model species, and it creates what's called a chain liftover file. So this tells you where one position in your model species is and where that is in your threatened species. And then that allows us to lift over these CAD scores onto the reference genome. So you now have CAD scores based for your reference genome, so that when you extract those the SNPs in your UCs, we can then get per-based CAD scores for every single site within those UC regions for your threatened species. So just give you a quick background, I know Hernán briefly touched over it on the conservation of the pink pigeon. So the pink pigeon is endemic to Mauritius and was originally found across the whole of the island of Mauritius, which is very beautiful if anyone's ever been there. And the population, as you said, went through a huge population decline to around 10 to 20 individuals in the wild in the 1990s. But before this, captive populations were actually started both on the island of Mauritius and globally in zoos around the world in the 1970s. And thanks to an incredible amount of conservation effort, now there are around 500 to 600 pink pigeons free living on the island of Mauritius itself, which, as Hernán said, led to the downlisting of the pink pigeon to vulnerable in 2020. However, as we touched on before, there was a lot of high levels of inbreeding going on in this recovery of this population. And this was covered in this paper that we published. As you can see, there's huge amounts of what are called runs of homozygosity, so areas where that genome is the same in those individuals, showing these high levels of inbreeding that were suffering through the population decline. And what we were able to do is perform vortex models to predict the future outcomes of the pink pigeon species. So we saw that if they just left the pink pigeon as it is with no demographic recovery at all, this is the black line, the population would go extinct within about 75 years. If we continue the demographic rescue that they're currently doing, we could make the population sustain until about 100 years in the future. But to truly get this population to sustain in the future, you need to do genetic rescue from those captive populations globally. So to give you a quick idea of this captive population, this is a very small subset of the pink pigeon pedigree, and this highlights the 6 individuals that we had sequence data for that we used for the publication. As you can see, there's already quite a bit of inbreeding that's going on within this captive pedigree. So to bring you back to those ultra-conserved elements that I said before, just to demonstrate where those are across the genome, so they aren't equally spread. There are some chromosomes that are enriched or chromosomes that are depleted for UCs. As we can see, those CAD scores in the flanking regions, so those are the red regions, are lower than the goes along, and then you get to the UC and you get this increase in CAD scores, goes back down, and you can see that split out where there's potentially a gene in that flanking region. So on average, the highest CAD scores are actually found within those flanking regions. So the most deleterious mutations, really highly deleterious mutations are removed from the UCs by selection. However, on average, CAD scores within the UCs are higher than those within the flanking regions. So you have a lot of these moderately deleterious or quite deleterious mutations, but not the most deleterious mutations within the entire genome. And another thing that we saw is that the actual number of these mutations appearing within the UCs is a lot lower than the flanking regions. This is as we would expect them being highly through the selection of being ultra-conserved. So what I was able to do is I was to take all of these CAD scores, and I would extract them for the UCs for every single individual and add these up to get those total genetic load that Hernán was mentioning, as well as the realized load and the mass load. So that realized load being the amount that's actually physically expressed within the individual, and the mass load being that hidden from selection. And although the total genetic load of the individuals is fairly similar across the population, that amount that is massed or realized within individuals, we could calculate and show there were slight differences between them. And then further to this, what I did was I simulated the potential crossings between every single individual, irrespective of the sex, to create what the potential offspring of those 2 individuals might be, and then calculate the amount of genetic load that would be present within those. And then when we did this, we could see that although the total amount of genetic load within the offspring didn't change with increased kinship, what we could actually see is, as Hernán was saying, that classical quantitative theory, that if you breed two individuals that are more closely related together, the amount of realized load is going to be higher. And we could actually show that here with the total amount of CAD scores between those individuals, that realized load being far higher in individuals that have higher kinship together. And what I was then able to do is I could then produce one of these correlation matrices. So here the blue squares mean that the offspring of those two potential crosses have a very low compared to all the other individuals amount of genetic load. And the more red a square is the higher amount of genetic load that cross has. So as you can see here, we could then calculate which individuals make a comparison between them. So you might highlight here this blue square here, but then when we split this out into the realized load and the mass load, you can see that actually that square that looked originally as quite a good cross to make is actually quite a bad one to do, because it would express a lot of realized load in the potential individuals of that cross. So we can then make really informed conservation breeding program decisions, because we can identify which are the potential best crosses to make for the future population of the species that we're investigating. What we wanted to do was also then validate this method that we designed within the pink pigeon and see how that worked on a species that was further derived from the chicken, and another species has been through a very similar situation. And that brings us to this project with the whooping crane conservation. So to quickly give an overview, the whooping crane is native to North America and is currently endangered. And very similar to the pink pigeon, it went through a massive population bottleneck, declined to around 16 individuals. And then captive bird populations were also started, including a migratory and a non-migratory population. And now, thanks to a lot of demographic rescue, this species has recovered to over 800 birds in the wild, shared across those populations, and they were able to perform captive releases from those captive populations back into the wild. Now, one of the great parts of this project is that a collaborator had already performed another method for identifying deleterious mutations called SNP-F. So this categorizes scores as either highly deleterious, moderately deleterious, or low likelihood of being deleterious. And I was able to show that those scores that we can get from CAD scores are very similar to those categories that you have in SNP-F. So if SNP-F categorizes a score as highly deleterious, we can show they actually do have very high CAD scores, whereas those that are predicted to be lowly deleterious do have low CAD scores within that individual. We were also able to identify quite a large number of sites that didn't actually get categorized by SNP-F, but as load lift lifts over every single site, we could give those a CAD score and see how much load was present within that individual. And what we were also able to do was then I could add up all of those CAD scores and calculate the amount of genetic load present within every individual, and could do that across these different samples that we had for the whooping crane. So we had individuals from the wild, the founding population, the early captive, and the late captive populations. And although we showed there wasn't a great difference in the genetic load or the realized load between those individual populations, what we showed across all of those populations is that the realized load is far higher than the mass load within these populations, which is kind of indicative of this huge population bottleneck that this species has gone through. And I was able to then also make those same simulated crosses that we did with the pink pigeons, and showing that with both increase in pedigree kinship, but also with genetic relatedness, you get that increase in the realized load within this population. So this brings me to the future applications of load lift and where I see it going. So load lift is currently available on GitHub as it came out with this publication that I mentioned before. To get advice on how to use it more, and if anyone wants to use it for anything, please do. So I see this really as a tool for assessing captive bird populations primarily. So where we've got a lot of that pedigree information, the life history of these individuals, we can make informed decisions to maintain these captive bird populations and ensure that they're surviving into the future so that we can really use them. And one of those uses as well is to screen these individuals from captive bird populations. If we are doing reintroductions into the wild, we want to make sure that we're not reintroducing any potentially deleterious mutations back into that wild population for these threatened species. And as Hernán mentioned before, we can also evaluate historical populations. So if we've got museum samples or long-term blood samples, we could then see how that genetic load changes over time. I'd like to thank all of these amazing people for all their very hard work and all their help with the development of load lift and the work, as well as all these organizations as well. Thank you very much.
All right. I think I can start. Okay. So it's my pleasure to share first work of our group on behalf of our colleagues. Okay. Is it working? Yes. So as we know that thanks to all those reference genome programs, now there are a lot of bird genomes, reference genomes online available. And I think so far we have almost 4,000 bird genomes that you can just download and use. And to make use of that, we want to put our malicious species into a scope of comparative genomics to see that on the concept of conservation genomes, how they behave compared to other species in a bigger scope. So that besides the pink pigeon I talked about, we have in our group, we have also been working on 2 other species. They have experienced similar demographic history, being into this extreme bottleneck. Mauritius kestrel, which went down to 4 individuals, and Comparative went down to 12 individuals. And they have all recovered more or less at the same time. And we have constructed their reference genome, very high quality, chromosome level. And then we picked 36 other bird species. We took their reference genomes, and then we used the raw rates, which used to assemble the reference genome and map the back so that we can get genetic parameters. And those species, we choose the high quality reference genomes, and then they're all across the bird phylogeny, and they have different conservation status, erosion status. And for that, we want to do to compare genetic and conservation status. Yes, sorry, it got a bit stuck. Okay, let's go back a bit. So for all the species, we estimated heterozygosity and then runs of zygosity. This is for inbreeding, as we said, and then we have adapted SAM's load lift pipeline to estimate the genetic load, and which is compared back to chicken. And we have also chose some falcon species and parrot species and some pigeon species just to compare within these taxonomic groups and to see their demographic history. And then we have also estimated genomic synteny and transposal elements, but this part I will not be talked about today. So we have seen that, yes, there's of course a huge range of heterozygosity among those certain species. We have our Mauritius species, so the Mauritius falcon lowest of all, and then we have the parrot still in the very low range, and then somehow the pink pigeon somewhere in the middle. This is a bit unexpected, but yeah, they have a big range. But the interesting thing that we don't really find a correlation between the conservation status and the heterozygosity. And for the runs-off zygosity, the FRH, so the percentage of the runs-off zygosity within the genome, it is roughly correlated with heterozygosity. The thing is that for some reference genomes, the individual that's used for the reference genome is a captive one or is a domestic one, which means it actually has higher inbreeding. So we have some noise. But again, it's roughly correlated, and FRH, which is also not correlated with erosion status. We further look into their demographic history, which was estimated using PSMC. So this graph here is from a closer time here is 5,000 years, and it dates back if you look into the x-axis. And then we use the harmonic mean between 10,000 years to 100,000 years. We use this as their historical effective population size. And then we took the modern census population size, how many you count today. Not today, but anyway. And then we log transform those numbers and then divided them. We get this NE-NC ratio, which you can sort of think that is how much it changes during time. And we have found this is actually correlated with the IUCN status, that if you have a higher NE-NC ratio, which means that they have declined during time, that is more likely that the species is in danger. And then we have also looked into the correlation between the historical NE and the heterozygosity fund is very closely correlated, which means that the heterozygosity we see today actually comes from a very long history. Remember that I used the historical NE from 10,000 to 100,000. That's quite a long time ago. And while heterozygosity is correlated, FRH is not, because as Hernán said, that it runs off homozygosity breaks down easily during recombination, so that it actually represents a way shorter history. And we also have looked into the genetic load. So here it's a bit different from a load lift. That is that because we have almost 40 species, and it all dates back to the chicken, so that we just excluded the substitutions that are shared among more than 20 species, because if it happens in so many species, it's likely to be a load. Rather, it should be more adaptive. Anyway, so here we have seen that for the heterozygous load, which is we use this to represent mask load, that it is correlated with heterozygosity. And okay, sorry, I forgot to change the text here, but you can just look at the figure. But homozygous loads is slightly correlates with heterozygosity, but it's not as strong as the heterozygous load. And as I said that because we compare from chicken, which is more or less at the very root of the tree, so the load we compare, it goes all the way from chicken to the terminal branches. So there is, it's not only the genetic load we are looking at here, but also there has to be some adaptive signals. So the homozygous load we look is not a real load, but it's a mixture of load and adaptive signature. It's more like a genetic background. And we also look between the three taxonomic groups we have, and we have seen that actually the genomic background between the groups can be quite different, because here we have heterozygosity, and the first one is falcons, and then parrots, and then pigeons. We can see that most falcons we have used in the study, they have a lower heterozygosity than pink pigeon. So the thing is that for when doing conservation genomics, it's better to look only within the groups, because when you look between groups, it's very different. And for FRH, they are more or less at the same level, roughly, and we have the endangered species and then the merged species. They are in higher level of their groups that expected. And then for the homozygous load, it's not really working. Okay, sorry, I get a bit stuck here. And for homozygous load, we also have seen that this genomic background is quite different between the groups. And for the demographic history, yes, again, for different groups, they have different demographic histories, and they for the falcons, most of the falcons has a relatively low historical effective population size that, as we said, it correlates with heterozygosity, so that could explain why they have a lower heterozygosity. And for the conclusions, first we have seen that the genetic background of the species is actually very affected by the long demographic history, and it is actually very useful for conservation. And this demographic history can be estimated just with one genome, the reference genome. And we have seen that species with recent decline are more at risk, which is kind of expected. And then we have seen like heterozygosity is more related to long history, while inbreeding estimates FROH is related to the short history. So they are estimating different things. And then we have seen that species with higher genetic diversity, usually we say that they have less risk, but when they decline, their the mass load can become realized load, and there's actually this risk for them. And we have also seen that different species groups can have very different backgrounds. This should be considered in genetics. And this work has been on bioRxiv. And if you're interested in more details, you can check out there. And I need to thank, of course, my group members and a lot of collaborators and all the help for the samples and the sequencing, and of course the phones. And of course, thank you for listening.
Okay, thank you so much. It's very outstanding and important work you are doing. So, for people seeing from YouTube, please post your questions in the chat. So, starting with Lucas Freitas, he has a question for Ana Morales. So, is it trustful to investigate runs of homozygosity metrics using rat-seq data?
Yeah, that's a very interesting question. I don't have a lot of experience with rat-seq data myself. I guess that it depends on how much you trust the genome and the density of the rat tags. I will, for example, in the Jackson et al. Paper that me and Sam show for the pink pigeon, we actually used rat-seq data and estimated runs of homozygosity, and we kind of like trusted it. And now that we're getting genomic data, they are very consistent, the measurements. We haven't checked if it's consistent at the raw level, but it's consistent at the FROH level. So I will trust it at that level. But I will say it's really, really dependent on the density of the rat tags. And as well, if you have a good reference genome, if you can infer the distance between tags across a long stretch of the genome. I don't know if Sam or Xuejing has any other thoughts.
Yeah, I think that.
Someone in the audience that has experience with that more than us. Well, okay.
So in the Lynette Rasmussen asked, I think it's for Samuel, so could the load lift be used for species that are not birds?
This is actually quite an interesting question, something we kind of want to look into. So we haven't used it ourselves on any species that aren't birds, but it definitely could be done for species that aren't birds. So there are, as I mentioned, CAD scores available for the human, for the pig, and for the mouse. So if you have a species that's fairly closely related to that and want to try it out, then yeah, you could definitely use the load lift pipeline for that. It's not really, we've designed it and tested it on the chicken CAD scores, but you could use it for the other species themselves. That would be interesting to see, and it should work.
Okay, in a follow-up question, what type of data does the load lift require in the quality of it? So it's all genome sequencing. How much coverage do you need for it to
That's a
work?
good question. The data, as I said, you require a reference genome. So for the pink pigeon that we applied this to, it wasn't a full chromosome level reference genome, but there is a new chromosome level reference genome you could do. So if you have a reference genome for your species, that's kind of one of the key things you need to extract those UCs, and read data for like your sequenced individuals that you have. That's kind of all you need is, and the other parts of it are included. Well, you would need to find the reference genome for the species that the model that you're lifting over from, and the categories. Yeah.
Okay, and how does it deal with degraded samples like museum samples that have very low coverage, like around 1x?
Yeah, we've tried it with some museum samples, and that is kind of one of the slight things that we've got as a consideration. So if it's more about that confidence of whether or not you've got a heterozygous or a homozygous site in that individual. That's where it comes into. Obviously, the load lift can say you've got different amounts of load depending on whether you've got that site is homozygous or heterozygous in that individual, and that affects those like mating crosses. But you really want to have good confidence that whether or not it is heterozygous or homozygous. So kind of that decreases with the obviously the decreased read depth that you've got. So
using low coverage data, could you use pool-seq? So if you have a population at low coverage, well, individuals at low coverage, but high coverage at population level.
Yeah, I mean, we have tried it, but that would be an interesting way to get around that issue. Yeah, that'd be.
Okay. So, Robert Waterhouse, so what is the preferred method for identifying ultra-conserved elements?
This is for me again.
Yes.
So in the low lift pipeline, we use a set of probe set that's produced the PhyLucy pipeline as part of the B10K data set. I think they released with that. So these identify just 120 base pair probes. So you could download them and run them in the pipeline. But if you had your own probe set, potentially for ultra-conserved elements for your species, then you could use that yourself. But yeah, this is designed for you input those probes that you say this is those ultra-conserved regions.
Thank you. So Alexander Zinenko, what about sex chromosomes and mitochondria, which have even smaller effective population sizes than the rest of the genome? Is the most vulnerable to drift part of the genome? Are there any data on that?
Yeah, thanks for the question. It's a very good one. Because usually for the conservation genomics studies, we only use autosomes because the sex chromosomes and mitochondria is a bit hard to deal with. I have some results that still in the ongoing work, but I can tell that we have observed for the malicious heteros that we have a much bigger loss of genetic diversity for the mitochondrial genome and the nuclear genome. And for the sex chromosomes, theoretically, you would expect, yes, it's more vulnerable because, for example, for the XY system, the Y is haploid all the time, and then the X is haploid diploid, which means that when it's haploid, it's completely exposed. So I don't recall seeing any studies. I think there are some theoretical studies, but yeah, that's definitely something we should look at.
I will add a little bit. So the mitochondria will be interesting as well, not to look necessarily at the deleterious variation because they are haploid genomes, so probably they express the deleterious variants differently. But these mitochondrial nuclear interactions, if they are disrupted, right? I think in our species, we go from few populations to one population, so probably there is not a lot going on. But if you have ancient population structure and different mitonuclear coadapted complexes, and you lose haplotypes more easily because they go through these bottlenecks, or they're affected through these bottlenecks more harshly, I will expect that the coadapted mitonuclear interactions get disrupted quite easily.
Okay. So Christian also has a question. So when estimating inbreeding levels from historical or ancient DNA, how can paleoenvironmental reconstructions such as past climate, habitat fragmentation be integrated into demographic and coalition models to improve inference of population structure and mating patterns over time?
Well, that's a tricky question. When estimating breeding levels from historical ancient, I mean, I don't know how far the sedimentary ancient DNA methods go as to estimate abundances, past abundances, if they can be linked in that way. But I know those methods are advancing quite quickly. And I imagine as well, like methods from modern environmental DNA trying to do PubGen can help there. So I imagine that there is future scope to investigate that. I also know some people integrate this modeling already. Maybe you can check out work by David Nogues Bravo and Richard Forham from Adelaide, and Elisabetta working with Forham. I think there is a lot going on at the moment, but we don't really work with the sedimentary ancient DNA in our group.
Okay. So another question from Robert Waterhouse. So we heard yesterday in the ERGA plenary that bird microchromosomes often don't get well and are therefore often missing from assemblies. So is this a major concern when looking for load in erosion?
I think for us it's more or less fine because depending on it really depends on like how the reference genome is assembled and the quality. And because for the malicious birds, we have reconstructed some of the microchromosomes, and also because like they're way shorter, they have less genes. So when we look at genetic load at the moment, we still only look at a very rough, big picture, which means that we just count. So that if it has less genes, we would expect that they carry less genetic load.
Yeah, I agree. At the moment, we compare genome-wide signatures of load diversity through time and between species. So not so much, but definitely when we start to dig in deeper into what happens across the genome, I think these things are going to start being very relevant. We're lucky to work with Don Matters gave the talk yesterday, so we're hoping to improve the microchromosome assembly parts for sure.
Okay, thank you. I don't think we have more questions. No. So I think we can close session for today. Thank you all three. It's an amazing work. I will send an email with more questions, especially to Samuel about your tool. And thank you all for attending the seminar on YouTube. Thank you all.
Thank you, Joao.
Thank you so much.
Thank you all
Thank you.
and Christian and Luisa. Thank you.
Thank you so much.
Thank you. Bye. Have a nice day.
Thank you.
Bye.